Hello, my name is Bradley Alicea. I'm from the Open Room Foundation and Orthogonal Research and Education Lab. And today I'm going to talk to you about physical intelligence and development, regulation of dynamic tension, and sensing as intersecting networks. The development of a complex organism typically involves two processes. The first is a series of differentiation events, and these are summarized in the tree at the right. This is a binary tree that has branching events throughout its topology and is organized in terms of developmental time. So the farther we go in developmental time, the more division events we have. The second process is the emergence of intersecting subnetworks. And so below you can see cell tracking data that are presented in 2 dimensions and 3 dimensions. These are newly differentiated neuronal cells in the C. elegans embryo, shown in the red and the blue. But we also have developmental cells or embryonic cells that are in the transparent color. So these two networks overlap and intersect. One way to think about this intersection is this concept of generative divergent integration. And we can use a toy model to understand how this emerges in development. So on the left, we have an 8-cell example, which is something that shows the emergence of something we call an embryo network. This embryo network emerges from cells based on their proximity to one another. And as you can see, at the 8-cell stage, you have these disconnected networks that only involve one cell type. At the right, we have the 24-cell example. And the 24-cell example has 2 cell types, and it's a little bit more crowded in terms of cells, so there's more connectivity. The network is continuous. But we also see the divergence of neuronal cells in green and developmental cells in blue. And some of these developmental cells will become somatic cells and turn into their own subnetworks that will eventually have less connectivity between different groups of cells, but the connection between different groups of cells will be maintained. So we have this simultaneous divergence of function, divergence of network topology, but we also have these sort of weak ties between different subnetworks. And so that's what we'll be talking about in more detail. So we'll consider the relationship specifically between tensegrity structures that form the generic animal body and the network of neuronal cells that exist within this generic animal body. So we're not going to be talking about any specific organism. We'll be talking about a generic organism as an example of this. This forms the connectome and forms the organism, the developing organism. As both of these networks emerge from an embryonic precursor, they have critical intersection points as shown in the divergent integration model in the last slide. So in this case, we see a very simple example of this. We have, say, for example, our tensegrity structure is represented by the 6-diagon and a hairball network, which represents the connectome, and those overlap in critical ways. Shown in the context of an organism on the right, we're a generic organism. We have this connectome in the front at the anterior end of the organism. We have a midline, and then we have these structures that are linked 6-diagons, which serve as this tensegrity network that give the organism stability and shape. So first top thing we have to cover is this idea of biological tensegrity. So biological tensegrity is not a new concept. It was somewhat popular in the 90s, biological tensegrity making the cover of Scientific American. We see at the left and on the right, we see cells. I think these are fibroblasts, and these cells have this structural integrity that has to be maintained as the cell changes shape and moves around. So you can see that there's a calculation of forces as the cell adheres itself to a surface and moves around freely. So this is all something that has to be done in real time. It's a dynamical process that we can actually use dynamical systems for. We can also use networks to understand the critical points of stability and then intersection with other networks. So this is an example of a tensegrity network. We talked about the 6-diagon already. This is a sphere. This is something that's stable, that's round, so it's biological in that sense. So we have that type of structure, but we also have other types of structures like the icosahedron or this 20-agon on the right. And this is something we'll actually work through how this network is characterized mathematically in a little bit, but we can see that we have these nodes, which are these structural pieces, and then these connections or these arcs, which are the things that introduce the tensions and loadings of forces. So you can see how this thing is held in stability by being organized in a way that allows us to have stability, stability of dynamic tension. So tensegrity networks are defined as dynamic tension introduced as a series of rigid and discrete compression elements joined together by continuous tension forces. So you can see that we have these, this network is discrete, but it's also rigid because it's held together through these forces. So the discrete components are the nodes, and the rigidity is introduced by the arcs and the forces along those arcs. So the network topology then leads to structures that are ultra-stable, resilient to shape deformations, and are highly modular. So this icosahedron on the right, as well as the 6-diagon on the left, are extremely stable structures. They can be modular, so they can be decomposed into different subcomponents, and they're resilient to different shape deformations, which we see a lot of in development. So in cells specifically, tensegrity networks can form a cytoskeletal configuration within and between cells, the interface of cytoskeletal elements being the site of force balance. So we can see a similar process in this figure. This is actually in an architectural structure where we have these components that are organized along an axis. They get put together in this sort of structure, and then they can be bent, and they can bend and undulate and all these other things. So if you, if you want to have a structure that's resilient over a number of transformations, then you want to have some sort of tensegrity network. So this is the icosahedron. This is the mathematical representation. We usually use a connectivity matrix. We denote D sub ij, and this is a way to represent this network as a series of connections. This will become important when we're looking at the intersection with connectomes a little bit later on. But these are discrete edges. So the edges are these lines, and the edges are either a 0, which is a passive force, or 1, which is an active force. The active force keeps these things in dynamic tension, but also sometimes at some points in time, there needs to be a passive force on that connection so that these nodes can change their position relative to one another. So as you can see, we have a 3-dimensional coordinate system, much like we had for the cells in the embryo, and those coordinates can change over time, and so do these connections between them, these edges. Biological edges are flexible over time, so they can have a value. Sometimes if you have a rigid structure, you want something that's either 0 or 1, but sometimes if you want some sort of flexible structure or soft material as the thing that's joining those things together, where the forces are being loaded, like you find in a lot of biological structures, you want to have a range of values between 0 and 1. Now we'll talk about the connectome and give different examples of a connectome. A lot of people are familiar with the hairball network. And the hairball network connectome is a connectome with a lot of connections. Some of them are ordered hierarchically, some of them are ordered somewhat randomly, and this is the type of connectivity we expect out of a connectome. This is an example of a generic hairball network, so it doesn't point to any one connectome. On the upper right, we have the labeled C. elegans connectome, which has actual C. elegans cells that are labeled according to nomenclature, and you can see the structure where it's a little bit more mature than the one shown in the first 2 slides, but this one has some structure going from head to tail and down the center of the body. So we can see that there's some anatomical structure to it. We also have the fly connectome, which has recently become available. The C. elegans connectome has been available for quite a while at the cellular level and actually at the connection level. So we know from which cell connects to which cell. But in flies, we also have this type of connectome where we can trace the connections between cells. And this is a larger number of cells and a larger number of connections. As you can see, it's still the same thing. You have this structure that's anatomically specific structure. So these are small animal connectomes are actually quite easy to work with. The reason I bring this slide up is because it's interesting the kind of data that's available. So in polychaetes, which are small worms, there are these very small nervous systems of under 100 neurons. and you can map those out and trace the connections between them. We also have other connectomes from other organisms, Platynersus, Siona, and C. elegans. And so all of those networks are available in different ways in the literature. So we can build these networks from the literature. We can build small networks as toy models and connect them with these tensegrity networks, which are, of course, available from anatomical data or can be constructed from just by simulating the shape of the organism. So the neuroconnectome consists of cells, of course, connected via electrochemical action, and maintains information processing between the body surface, the periphery, and the central nervous system. So we can see at the upper left, we have an example of this nervous system. We have the cells and we have the nerves that come out, and we have this conforming to an adult. This is a C. elegans body. So that's what we can look for in terms of the connectome being connected to the rest of the body and being embodied in that way. And then at the bottom left, we have this map of a navigation circuit, which is a smaller subset of the connectome. And the reason I bring this slide up is because I want to show where those things are with respect to the body and with respect to behavior, because the organism is behaving, and you can see that a majority of the circuit is towards the head, towards the anterior end. And so this is where a lot of this action of the circuit takes place. And so you can see we can represent a connectome with this other matrix, which it looks a lot like D sub ij, but this is C sub ij, and this is just the connectivity, whether it's positive or negative from one cell to another. So this is the navigation circuit. I have, I think, 11 cells by 11 cells in this matrix, and I have positive and negative marks. So those two, we have both of those types of data that are available as matrices. We can then look at the intersection of these matrices, which we'll talk about in a little bit, and get a better sense of how those things work. I want to bring up another topic, which is the idea of embodied networks and hypergraphs. So this is a type of network that's embodied, much as like our connectome, and it goes from some sort of input, some sort of sensory input, to an output. And so put aside what we know about connectomes for now, and just think about the path between a sensory organ or a sensory cell and an effector cell, which could be an effector organ, something like a fin or maybe like muscles. And so we have these paths between the input and output, basically, and we can embody these like we did with the connectome in the previous cases, and we can also have signals that pass from the input to the output. Now, this is a very simple example of an embodied network, but this is what we want to get. We want to move towards a little bit more complex model of this. So we can actually introduce interneurons, which are processing units in between the input and output. Sometimes they summarize different paths of information. Sometimes they disperse them across the number of cells or interneurons. And so this is a way we can build a more complex network that's embodied, and that maybe can actually do some sort of behavior. So these are informational relays that can go from input to output. And so we can embody these networks in that way. Now we can also not just use them as networks, but also a hypergraph. So in the case of hypergraphs, we have nodes that have a number of cells within them, and they have a diverse set of properties, and they also have a diverse set of connectivities that we can also summarize. But in the hypergraph case here, we have the ability to characterize sets of cells that contribute to, say, like an organ or a tissue, and use that as a single node. So we can actually do some really interesting things with that. And so we can actually orient these networks towards behavioral or sensory inputs and behavioral outputs. And we've talked more about this in this preprint listed here. And each hypergraph node is a structure or tissue type, and it generates a spectral graph of cell type properties. So at the end of the day, we can take each one of these nodes and get cell type properties out of them. This may describe multiple scales of differentiation and precursors to phenotypic change. But what it can also do is it can describe its relationship to this other network, this tensegrity network, and it can interact with that network. So this is an example of where we are. We have the connectome, we have this tensegrity network. It's organized in an embodied manner from anterior to posterior end in a left to right symmetry with a midline. Now, this is the interesting part when these two networks intersect. So we're connectome C sub ij and our tensegrity network D sub ij intersect in different ways. And this is just showing a very simple case where these two parts of the network or the matrix intersect. And we can see this in biological systems. And these intersections come in the form of something like a proprioceptor. So this example is of a skin proprioceptor and a skin nociceptor. And so this shows examples of these different sensory organs at the surface of the body. And the body, of course, is being held in rigidity, and there's information processing going from the surface of the body into this nervous system, and then it goes on to generate behavior. So you can see that there's this network that comes out of the surface and down into an internal connectome, and it's generating behavior, which in turn affects the structure, the stability of the tensegrity network. So this is an example of an overlay in the adult of these two different intersecting networks. This is in C. elegans, this is the somatic and the nervous system network. So you can see that the somatic network, made up of a cuticle and of muscle, is overlaying this nervous system network. And you can imagine that there's some sort of sensory input at the side of the worm or at the front of the worm, and it's being transduced into the worm's nervous system, and then that nervous system causes behavior, and that affects the stability of the organism. It affects the stability of the internal network, and so forth. So we can see as this changes its perspective, that you get different views of things, and you can see how that those two structures interact physically. But we can also look at this in terms of a systems context. So there are proprioceptors around the left. You have an input of sensation. There are transduced forces that come into the organism. And these transduced forces come through the tensegrity network. So these cells here represent a loosely organized tensegrity network. They can shift their position as there is growth, as there's regeneration, as there's movement of the organism. And those proprioceptors are just inputting a lot of sensory information that gets transduced. But of course, then we also have our connectome, and the connectome is sending signals out, especially as it's starting to develop and form connections with the neural cells. So as I told you before, there is this divergent integration. Some of this integration comes in this form, where you get these interactions between the structure of the body, the sensory inputs, and the outputs of a connectome. In addition, there's cell growth and shape deformation, and a response to forces, which is action from this emerging connectome. What's interesting is that we expect that in development, these interactions move from being cell autonomous or reflexive to goal-directed, or being the product of the connectome entirely. So in development, the cells move around. They have a lot of motility in the embryo. And then later you have muscles that sort of twitch without being connected to anything. And then eventually the connectome is actually managing a lot of these interactions. So it's a very interesting set of interactions going on there. So we can actually go beyond the linear case. We can look at a number of examples from developmental biology and start to think about how those things are to be reinterpreted in light of what we've just discussed. So for example, there are things like furrow formation is invagination, which is the folding of a cellular sheet inward. We have contractility and ratcheting, where the bean or the oblong mass that's the embryo stretches out and folds. We have pulsitations and flows, which are different ways that sheets of soft materials, of which our cells are part of, change their shape due to physical forces. We have caveoli and doline formation, or nanoevaginations, which are the same as your furrow formation, but this occurs on the surface of cells, and it's at a much smaller scale. And finally, push-pull reciprocity, where there's deformation of a sheet and will fold or into a nodule. And so all these things are this sort of morphogenesis that involve tensegrity network, but also signals that come from maybe a centralized information processing place, or sometimes cells acting collectively. Finally, we can look at this from a broader lens as what they call 4E cognition. So we can look at this in terms of embodiment, but also an action, embeddedness, and extended cognition. And we don't think that embryos exhibit cognition, but we can look at information processing using some of the things from this literature. Thank you for your attention.